Good day. I'm Louise. So for today, I'm going to discuss glycolysis. What is glycolysis? Glycolysis comes from a merger of two Greek words, glyx equals sweet, lysis equals breakdown splitting. It is also known as Embden-Merhoff-Parnas pathway or EMP pathway. Also, glycolysis is the process in which glucose is broken down to produce energy. It makes two molecules of pyruvate, ATP, NAD, and water. The process occurs in the cytosol of the cell cytoplasm, the presence or absence of oxygen. Glycolysis is the primary step of cellular respiration. Two phases of glycolysis. Glycolysis leads to the breakdown of 6Z-glucose into two molecules of 3C-pyruvate, with the enzyme-catalyzed reactions being bifurcated or categorized into two phases. 1. Phase 1 preparatory phase. 2. Phase 2 payoff phase. Glycolysis is the sequence of 10 enzyme-catalyzed reactions that converts glucose into pyruvate with the simultaneous production of ATP. The overall reaction of glycolysis which occurs in the cytoplasm is represented simply as C6H12O6 plus 2 NAD plus 2, and orin pesadas plus 2 P2 pyruvic acid, CH3C equals OQ plus 2, ATP plus 2, NAD plus 2H plus. Remember, glycolysis is the first step in the breakdown of glucose to extract energy for cellular metabolism. Nearly all living organisms carry out glycolysis as part of their metabolism. The process does not use oxygen and is, therefore, anaerobic. Glycolysis takes place in the cytoplasm of both prokaryotic and eukaryotic cells. That's all. I hope that you learned something. Thank you.
Hello. My name is Jonasis and I'm doing a report about Krebs cycle. Krebs cycle is the cycle of chemical reactions that are the major source of energy in living organisms. Krebs cycle. The Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle, is one of the most important reaction sequences in biochemistry. Not only is this series of reactions responsible for most of the energy needs in complex organisms, the molecules that are produced in these reactions can be used as building blocks for a large number of important processes, including the synthesis of fatty acids, steroids, cholesterol, amino acids for building proteins, and the purines and pyrimidines used in the synthesis of DNA. TCA cycle supplies energy and also provides many intermediates required for the synthesis of amino acids, glucose, heme, etc. TCA cycle is the most important central pathway connecting almost all the individual metabolic pathways. Reactions of TCA cycle. Oxidative decarboxylation of pyruvate to acetyl-CoA by PDH complex. This step is connecting link between glycolysis and TCA cycle. Step 1 formation of citrate. Oxaloacetate condenses with acetyl-CoA to form citrate, catalyzed by the enzyme citrate synthase. Inhibited by ATP, NAD, citrate competitive inhibitor of oxaloacetate. Steps 2 and 3 citrate is isomerized to isocitrate. Citrate is isomerized to isocitrate by the enzyme aconitis. This is achieved in a two-stage reaction of dehydration followed by hydration through the formation of an intermediate cis-aconus. Steps 4 and 5 formation of a cataglutarate. Isocitrate dehydrogenase, ICDH, catalyzes the conversion of oxidative decarboxylation of isocitrate to oxalosuccinate and then to a cataglutarate. The formation of NAD and the liberation of CO2 occur at this stage. Stimulated, cooperative, by isocitrate, NAD+, Mg2+, and orin pesadas, Ca2+, links with contraction. Inhibited by NAD and ATP. Step 6 conversion of a cataglutarate to succinyl-CoA. Occurs through oxidative decarboxylation, catalyzed by a cataglutarate dehydrogenase complex. A cataglutarate dehydrogenase is a multi-enzyme complex. At this stage of TCA cycle, second NAD is produced and the second CO2 is liberated. Step 7 formation of succinate. Succinyl-CoA is converted to succinate by succinate thiokinase. This reaction is coupled with the phosphorylation of GDP to GTP. This is a substrate-level phosphorylation. GTP is converted to ATP by the enzyme nucleoside diphosphate kinase. Step 8 Conversion of succinate to fumarate. Succinate is oxidized by succinate dehydrogenase to fumarate. This reaction results in the production of FADH2. Step 9 Formation of malate. The enzyme fumarase catalyzes the conversion of fumarate to malate with the addition of H2O. Step 10 Conversion of malate to oxaloacetate. Malate is then oxidized to oxaloacetate by malate dehydrogenase. The third and final synthesis of NAD occurs at this stage. The oxaloacetate is regenerated, which can combine with another molecule of acetyl-CoA and continue the cycle. This is the Krebs cycle looks like. Regeneration of oxaloacetate. The TCA cycle basically involves the oxidation of acetyl-CoA to CO2 with the simultaneous regeneration of oxaloacetate. There is no net consumption of oxaloacetate or any other intermediate in the cycle. Significance of TCA cycle. Complete oxidation of acetyl-CoA. ATP generation. Final common oxidative pathway. Integration of major metabolic pathways. Fat is burned on the wick of carbohydrates. Excess carbohydrates are converted as neutral fat. No net synthesis of carbohydrates from fat. Carbon skeleton of amino acids finally enter the TCA cycle. Requirement of O2 by TCA cycle. There is no direct participation of O2 in TCA cycle. Operates only under aerobic conditions. This is due to NAD+ and FAD required for the operation of the cycle can be regenerated in the respiratory chain only in presence of O2. Therefore, citric acid cycle is strictly aerobic. Energetics of TCA cycle. Oxidation of 3-NAD by ETC coupled with oxidative phosphorylation results in the synthesis of 9-ATP. FADH2 leads to the formation of 2-ATP. 1 substrate level phosphorylation. Thus, a total of 12 ATP are produced from 1 acetyl-CoA. Transamination is a process where an amino acid transfers its amino group to a keto group and itself gets converted to a keto acid. The formation of alpha-ketoglutarate and oxaloacetate occurs by this mechanism. 3 regulatory enzymes. 1. Citrate synthase. 2. Isocitrate dehydrogenase. 3. Acetoglutarate dehydrogenase.
Hello, I am Bianca.
Hi, I'm Cyrus.
And we're going to discuss the electron transport chain.
The electron transport chain is a cluster of proteins that transfer electrons through a membrane within mitochondria to form a gradient of protons that drives the creation of adenosine triphosphate, ATP. ATP is used by the cell as the energy for metabolic processes for cellular functions.
Electron transport chain. ETC is the fourth and final stage of aerobic respiration. Through ETC, the E needed for the cellular activities is released in the form of ATP. ETC is an O2-dependent process which occurs in the inner mitochondrial membrane.
The electron transport chain is a series of 4 protein complexes that couple redox reactions, creating an electrochemical gradient that leads to ATP's creation in a complete system named oxidative phosphorylation. It occurs in mitochondria in both cellular respiration and photosynthesis. The 3 main steps in the electron transport chain. Step 1. Generating a proton motive force. Step 2. ATP synthesis via kinesimosis. Step 3. Reduction of oxygen. Summary. Oxidative phosphorylation.
When no oxygen is present, the electron transport chain can't run because there is no oxygen to act as the final electron acceptor. Both glycolysis and the citric acid cycle require NAD+ to accept electrons during their chemical reactions. Location ETC is located in mitochondria, the centers for metabolic oxidative reactions to generate reduced coenzymes, NAD and FAD, which in turn are utilized in ETC to liberate E in the form of ATP. Hence, MC is regarded as powerhouse of the cell. That's all. Thank you.
We hope that you learned something.